All right, let's jump right in. We are about to walk through one of the most important scientific papers ever published. I mean, seriously. Back in 1953, in just over one page, James Watson and Francis Crick laid out a structure for a molecule that would completely change the world. This is the story of how they unlocked the secret of life.
And it all started with this sentence. Talk about an understatement, right? No big fanfare, no dramatic claims, just a simple suggestion. But that one little sentence kicked off a revolution in biology that we're still feeling today.
Now, to really get how brilliant their model was, you've got to understand what was happening at the time. See, Watson and Crick weren't working in some kind of bubble. There was a full-on race to figure out the structure of DNA, and some of the biggest brains in science were on the case.
People were getting close. I mean, you had giants like Linus Pauling and Robert Corey who had already published their own model. There was another one from a scientist named Fraser. All the puzzle pieces were kind of sitting there on the table, but as Watson and Crick pointed out, the way people were putting them together, it just didn't work. They were, in their own words, unsatisfactory. So what was the problem with the leading theory from Pauling and Corey? Well, their model had 3 chains all twisted up, but they put the phosphate groups on the inside, right near the center. Watson and Crick spotted 2 massive problems with that. First off, phosphates are negatively charged. Packing them that close together in the middle would be like trying to force the negative ends of 3 super strong magnets together. They just push each other apart. The whole thing would be unstable. And second, when they ran the numbers, some of the distances between the atoms were just physically impossible. The model was literally too crowded to exist. And this is where Watson and Crick made their move. They didn't just try to fix the other models. No, they basically tossed them aside and came up with something fresh, something bold, and something that turned out to be absolutely right.
You gotta love the confidence in this line from their paper. Not just different, radically different. They knew they had something big. Their idea basically flipped the old models inside out, and in doing so, solved all the problems that had everyone else stuck. Okay, so here's the core of their idea, and it's just so elegant. First, forget 3 chains. There are just 2, and they're coiled around each other. Second, and this is the game changer, they put those negatively charged phosphate backbones on the outside, which completely solves that repulsion problem. And third, that means all the important stuff, the chemical bases that actually carry the genetic code, they get tucked away safely on the inside, facing each other. And here it is. This is the actual drawing from their 1953 paper. It's so simple, but it's all there. You can see the 2 ribbons on the outside. Those are the sugar-phosphate backbones. And the horizontal lines? Those are the pairs of bases connecting the 2 sides, kind of like the rungs on a twisted ladder. This right here is the birth of the iconic double helix. They also got incredibly specific with the details. The 2 chains are what we call anti-parallel, which just means they run in opposite directions, like 2 lanes of traffic on a highway. The twist is always right-handed, like a normal screw. And the geometry is super precise. The whole thing makes 1 full turn every 10 bases. Oh, and putting those phosphates on the outside had another cool benefit. It meant positively charged ions could get in there and help keep the whole molecule stable. But here comes the masterstroke. The shape was brilliant, sure. But the real secrets, the part they called the novel feature, was all about how those rungs of the ladder, the bases on the inside, actually connect to each other. What they figured out was that the pairing couldn't be random. If the ladder was going to have a nice, uniform width all the way down, then a big base, which is called a purine, always had to pair up with a small base, a pyrimidine. You see, you couldn't have 2 big ones together or 2 small ones. It had to be one of each to fit just perfectly. And the rule gets even more specific than that. It wasn't just any big one with any small one. Because of their chemical structures, they figured out that adenine, or A, could only bond with thymine, or T, and guanine, G, could only hook up with cytosine, C. That's it. A always with T, G always with C. This is the fundamental rule that runs all of life as we know it. And that simple rule has a mind-blowing consequence, which they spell out right here. Think about it. If you know the sequence of bases on one side of the ladder, let's say it's AGGTC, then you automatically know the sequence on the other side. It has to be TCCAG. The 2 strands aren't identical at all. They're complementary. Each one is a perfect mirror image, a template for the other. And that idea right there, well, that leads to the absolutely breathtaking conclusion of their paper. This structure wasn't just some static chemical blueprint. The way it was built actually suggested how it could copy itself. You see, if each strand is a template for the other, you can just imagine the whole thing unzipping right down the middle. Then, each of those separated strands could be used as a pattern to build a brand new partner, and the result would be two perfect copies of the original DNA. The structure literally contained the secret to its own replication.
This line, "This is without a doubt one of the most famous and coolest understatements in the entire history of science." With this one, almost casual sounding sentence, they basically say, oh, and by the way, we think we've just figured out heredity. They didn't just find a structure, they found the mechanism of life itself.
And that leaves us with this final, just awe-inspiring thought. The unbelievable diversity of all life on this planet, from a tiny bacterium to a giant blue whale, from a mushroom to you and me, it's all written in a simple 4-letter code of A, T, C, and G, held together in this beautiful twisted ladder. The question isn't just how they figured it out, but how can something so simple contain all the breathtaking complexity of life itself?